Hello everyone. Today I would be discussing about green and sustainable water purification membranes. We are from UNC Charlotte, North Carolina, and we are supervised by Professor Jordan C. Polar. Let me first introduce you to my university. It is a host for multicultural background and interdisciplinary collaborations. It has wide range of clubs and activities for students to engage in. For example, our famous 49er teams. It also shapes the overall growth of individual by offering courses and training related to lots of technical and interpersonal skills apart from regular course curricula. Now let's focus on the problem that we are trying to solve here. There are several tiny molecules that are present in drinking water that we consume every single day. They are extremely deleterious to human health and environment, and they are very difficult to be removed from water and needs advanced technologies to do so. Hence, we are focusing on removal of these tiny molecules from drinking water and hence purify water as opposed to filtrating water that is focused in third world countries. There are various classes of contaminants that I am talking about, which ranges from PFAS or perfluoroalkylated substances that are found in furniture, cookwares, etc., to consumer products, antibiotics, disinfection byproducts, pesticides, etc. Then this is the information from health advisories that are established by US EPA, which defines the maximum concentration limit or toxic level of these kind of contaminants. The lower the MCL values, the higher is the toxicity. For some molecules like PFAS, as you can see, the MCL value goes as low as 70 parts per trillion, which says how pervasive these molecules have been in the environment. There are several advanced water purification techniques to remove these hydrophilic molecules from water that include photocatalytic oxidation, flocculation, coagulation, adsorption, ion exchange, and membrane technologies. We have been taking advantage of properties of anion exchange resins and integrating them with nanomaterials to formulate not only fast and high capacity nanoadsorbents but also sustainable and green um, water filters that could be regeneratable. The inspiration of our P3 project comes from our previous research work where we use the procedure of ARGIT ADRP, which is activator regenerated electron transfer atom transfer radical polymerization, to grow anion exchange resins in the form of controlled linear polymers. Every monomeric unit of these polymer acts as an active binding site to the contaminant molecule. In this case, the carbon nanotubes and the grown polymer, they form strong covalent bonds and these polymers hang on the surfaces of carbon nanotube as brush polymers. This work has been done previously where we did not require any organic solvents and used green processability to form these aqueous nanoresin, which can remove or absorb these kind of contaminant molecules very easily and in a fast process. But carbon nanotube is expensive in nature, and that is why we did the technology transfer to cellulose nanofibers. Hence, as discussed, even though carbon nanotubes paves the platform for green sustainable synthesis of nanocomposites, which render fast and high capacity adsorption, they limit the cost or affordability options. Hence, the technology transfer was done to cellulose nanofibers, which are plant-based, carbon neutral, affordable, inexpensive, and they have this carbon 6 hydroxy group functionalization possible, and they adhere to green chemistry, and hence we can synthesize sustainable nanomaterials, nanocomposites, and they also have, and our end goal is also to have regeneratable, fast and high capacity nanocomposites out of cellulose nanofibers. In this slide, we are talking about technology transfer process involved. Cellulose cannot be directly functionalized to polyelectrolyte like carbon nanotubes, and hence this is what we do to attain the functionalization. We convert the C6 hydroxy group to C6 carboxy group using the tempo sodium bromide and sodium hypochlorite method. In this process, while conversion or while doing the surface modification, we also introduce sonication so that we can debundle the cellulose fibrils to individual cellulose strands. And also because of the surface modification to carboxy groups, it forms a stable dispersion of celluronic acid. The C6 carboxylate groups then react with allylamine hydrochloride in presence of EDC to form stable amide bonds. This leaves vinyl group hanging on the surfaces of cellulose fibers, and that helps in the covalent attachment of polyelectrolyte. Then we grow the polyelectrolyte with the help of rjet ATRP and then functionalize it on the surface of cellulose fibers and form nanocomposites of combination of anion exchange reagents with the cellulose fibers. This is just pictorial representation of how the conversion happens. We have towards the left side, we have the cellulose fibrils that under the chemical treatment, we convert into celluronic acid, and upon sonication, we exfoliate these individual fibers and also surface modify it to celluronic acid of C6 carboxylate groups. The supernatant obtained of carboxy-modified celluronic acid, we combine it with EDC and allylamine hydrochloride to form stable amide bonds that is shown in the second picture, and then we functionalize it with polymer and it turns into this white creamy solution dispersion, and upon purification we obtain the polyelectrolyte covalently functionalized on cellulose nanofibers, and we call it PCN. Moving on to the results section, this slide represents the dynamic light scattering experiment where we compare the hydrodynamic diameter of cellulosic nanofibers versus celluronic acid nanofibers. The bundling of cellulosic nanofibers in aqueous dispersion gives rise to a size distribution of 259 nanometers as opposed to the exfoliation under sonication of celluronic acid. It gives rise to individual fibers as 24 nanometer size distribution. This slide represents the ATR FTIR of commercial cellulose as it has been shown in the spectra. The absorption band at 3331 centimeter inverse, it pertains to hydroxy groups present in the glucose units. 2890 centimeter inverse and 1315 centimeter inverse, it pertains to stretching and deformation bands that is present in the CH groups of glucose unit. Then the signal at 1037 centimeter inverse, it is assigned to the CO groups that is present in the secondary alcohols and ethers in cellulose chain backbone. This slide represents the ATR FTIR of amide surface modified cellulose. Here we can see the appearance of peaks pertaining to the amide bonds, especially the 1640 centimeter inverse pertaining to CO stretch, 1535 centimeter inverse pertaining to in plane NH bend, and 1429 centimeter inverse and 1260 centimeter inverse pertaining to CN stretch. Again, which reiterates the successful surface modification with secondary amide bonds in cellulose nanofibers. This slide compares the zeta potential of celluronic acid, amide surface modified cellulosic nanofibers, and polyelectrolyte functionalized cellulosic nanofibers. As shown in the slide, the zeta potential of celluronic acid is negative, and so is for amide modified cellulosic nanofibers. But the zeta potential of polyelectrolyte functionalized cellulosic nanofibers is positive. It is because the anion exchange reagents are positively charged molecules that when surface functionalized give a positive zeta potential to the cellulosic nanofibers. We perform water purification through membrane system. Towards the left, I have shown a simulation of fast purification setup. At the top, we have sodium fluorescein solution, which is colored, and it will help us to quickly analyze the filtration process, as under a positive pressure, contaminant solution is flown through the film of polymer brush coated cellulose nanofibers. And as we see, the filtrate is coming out to be clean and clear. Towards the right at the top, we see the PCN film that is deposited on mixed ester cellulose membrane, and at the bottom we see the PCN film is coated, it's yellowish because it has absorbed all the sodium fluorescein that was flowing through, and hence it looks yellow due to the anion exchange reason that absorb all these contaminant molecules. We have performed preliminary data analysis on adsorption profile of PCN. Towards the left, I have shown adsorption kinetic data, which shows the maximum adsorption being achieved within just few seconds. This is because PCN is open contact resin, which allows all the active sites to be already exposed for adsorption, and hence the pseudo equilibrium is achieved really fast. Towards the right, we have the adsorption loading analyzed as a function of equilibrium concentration. Here, the curve was fit using Langmuir and Freundlich models, and according to AIC test, Freundlich seemed a better fit compared to Langmuir model, and the absorption capacity, or KF, value came out to be 14.4, which is significantly greater than the graphene oxide membranes, which are the counterparts used for water purification. Lastly, this data is important because I have been talking about cost and affordability options that is limited in carbon nanotubes due to the high cost of the raw material. For cellulose, it is inexpensive that way, but all the nanoresins that we synthesize in our labs are regeneratable, reusable, and hence they are sustainable and long lasting, which is required. And also in this graph, the cellulose membranes are able to retain the capacity at the same level even after 37 cycles, with very less decrease in slope per cycle. This makes these membranes really fast, affordable, and high capacity nanocomposite for water purification applications. At last, we also want to discuss the preliminary ideation of fabricating prototype. The crude idea is represented in the leftmost, where we have a unidirectional flow tube with two segments of filtration. As water flows through the housing, it first goes through a robust anion exchange functionalized zeolite system where it removes all the bigger contaminants, and as the water is filled in the outer chamber, that water exudes through the inner perforated tube that is wrapped by a filter membrane that is functionalized with PCN. It can capture the tiny contaminants like PFAS as we have discussed before. In the center, we have a membrane filter that is coated with multi-wall carbon nanotube that is fabricated in our lab. Towards the right, we are showing a setup of fabrication of nanocomposites in the inner walls of the tube that is wrapped around by porous filter paper, and we are still working on the coating process in order to form a fully functional water purifier. We also try to increase the outreach and educational aspect of our group, and hence we took our group to a Mallard Creek wastewater treatment plant. It was before COVID, and these are some pictures where we understood how the wastewater treatment process happens in public utilities, and this was really helpful. And we would have also gone to uh, drinking water treatment facilities, but again the COVID happened, so we could not go there. In conclusion, I would like to say we have successfully synthesized polyelectrolyte or short strand anion exchange resin functionalized cellulose nanofibers with high density functionalization and using mild conditions adhering to green chemistry principles. According to the adsorption kinetics results, we observe that PCN offer facile, high capacity, and fast adsorption. Initially, I was discussing about MCL of emerging contaminants. Our next step includes testing removal of these emerging contaminants at that concentration range and seeing the percentage removal by PCN. We also need to analyze the structural aspects of these nano composites in order to understand the functions better, and we would be doing that using electron microscopy techniques like SEM and high resolution TEM. We are also planning on putting together a manuscript by consolidating all the data that we have and also gathering all the future data. We are also planning to collaborate with the local chemical company, Goulston, and further work on the fabrication of fully functional, affordable membrane filters. I would like to thank entire Polar Research Group who was involved in EPA P3 Phase 1 grant. You all have been phenomenal in helping this project to succeed in this pandemic. I would like to especially thank Professor Jordan C. Polar, who has been very protective and supportive to all these students during this pandemic situation. I would like to thank US EPA for providing us with Phase 1 grant funding and offering us such a prestigious platform to disseminate our novel work. Thank you all for attending the presentation and would be happy to take questions at Projects Comment page. Thank you.